Okay, so we're going to start by making a new Jupyter notebook. So it'll be Python 3. And then what I'm going to do is paste in the libraries that I'm going to use. So I've already prepared this, and I'll just explain what each one is and why we need it. So matplotlib inline, this is a magic function which is, as I believe, unique to Jupyter notebooks, and this represents a collection of functions for plotting. And this means inline will mean that any plots that we produce will be in the notebook and won't produce a separate figure. And then we import, as a convenience function, pylab as plt, so that we can do things like imshow and plot. And these will be displayed in line with the above. And then we want to be able to read our files. So in this case, our files are as TIFF files, so I use the library TIFF file. And in the description for this video, I will put all of the requirements and how best to install them. Some are easier than others. TIFF file can be installed by just pip. And then we're going to use NumPy, and I always do NumPy as MP. And then we need skimage library, which is good for analyzing and processing images, scikit-image. And then we also want n-dimensional image from SciPy, and we want the morphology filters as well. And also from scikit-image, we want to use flood fill, and also Watershed from the morphology library. Now this one, Find-maxima, is a module made by myself, and I will, in the description, put a link to how to install that, but it can be done through just a few simple steps. And then finally, we're going to use pandas. Now this function here, this is the only sort of function that we need to write ourselves for this pipeline, and you can just copy this from the GitHub, which I'll link to, but it's essentially what it does is it's a filtering step to reveal the local maxima in an image, and as we'll go through, you'll see why we need that. So the first thing to do is just to run that cell, make sure that you get a number here and no errors, so all of those libraries have been imported correctly. Obviously, if you have a problem there, then you'll have to address which of the modules hasn't been loaded and then install it before running again. So now we're going to import our images. So I'll just write a comment, import images, and then the files as they've been supplied in this example represent a separate file for each channel of the image. So we're just going to do first channel, and then we're going to use imread, which we've imported from TIFF file, and then we're going to import, and I'm using tab to automatically find the folder within the directory where I've placed the images, and then I'm going to select from the list which one I want to do. So we're going to start with 435 as the wavelength. I'll just expand this so we can see it. And then we're going to import the second channel, call it ch1, read, and then let's do it like this. And then the 676 channel. So I've chosen the wavelengths here represented by these numbers, and I've just made sure that they're in sequence. Okay, so we can then import them. They've all imported, and then what I want to do is just have a quick look at them. So I'm just going to create a figure using the PLT library. I'll make it quite big so we can see it. And then do a subplot so that we can display it as a row of images. So we're just going to put it in the first one, do the second one, done all of this right, then they should magically appear as a row. Oops, there's a spelling typo. So here we can see in channel 1, channel 0 even, our DAPI image, which we'll use for demarcating the cells. And then you can hopefully see here the bright blobs, the foci, represent our fish fluorescence in situ hybridization stained DNA sequences, which we want to co-localize between these two channels. So now we've got the data in, we can start doing some processing. And what we're going to do is first we want to identify our cells as best we can as individual cells. So we're going to threshold and then do some labeling to separate those cells. So for this we're going to calculate a threshold based on the histogram, intensity histogram of the first channel using an algorithm called UTSU. So this will look at all of the intensity values within the image and range them as a histogram, and then try and find a level, a value which best separates the background from the foreground. So what we first do is calculate the threshold using that first line, and then we're going to apply that to our image by just using a classic inequality based on the value that outputs. And then we can do im_show. So all of the pixels above this fresh value will be foreground and the rest will be background. And then we'll do an im_show, and I've done a typo there, so just change that. Have I spelt it wrong? I have. There we go. So you can see this has split this image between foreground and background relatively nicely. Now we can see that quite a few of the cells have been joined up as one. So if we were to try and separate this into individuals, we'd run into problems because these large blobs represent multiple cells. So what we want to do is try and look at the morphology and separate these cells into meaningful regions. And so one way of doing that is to do a watershedding procedure. So what we want to do first of all is try and identify the cells as best we can. Now a good way of doing that is actually to use a similar strategy to what we're going to do to find the foci, which is really to find the bright areas within the image and then try and separate them. So what we're going to do is we're going to represent these cells in a different way. We're going to process them so that the centers of each cell represent the peaks in the image, and then we're going to try and identify those peaks and then separate these areas based on the cell centers as best we can. So what we're going to do this is first we're going to apply a distance transform to our image. So I'll apply this, and then I'll visualize what it looks like. So distance underscore transform, and this is from the n-dimensional image library. So hopefully you can see now, in comparison to the above image, there are now peaks which represent the center of the cells. And then what we can do is we can identify these peaks and use it to separate these cells which were joined up in the binary. It's like a neat trick to do that. Now, to do that, I mentioned that I would use the same maxima finding algorithm which I'll use later. So this is a neat algorithm which I wrote in Python, but I've taken it from someone else originated the algorithm in Java, and I've taken it and I've applied it in Python. So I use this a lot for finding local maxima in an image. So this isn't a global maxima, this is bright blobs in relation to surrounding pixels in an image, and it has one parameter which is a noise tolerance. and I'll set that to 15. And then what we want to do is we want to make sure that our image is scaled between 0 and 255. So what I haven't done yet, and I probably should do, is convert these to a floating point bit depth. This is just something which means that if you import the images, they may be in a certain bit depth, but if you convert to a floating point, then it's basically saying that when I do division and things on these image, then the bit depth, the data type of the pixels will accommodate any kind of divisions and multiplications that I do, rather than doing some rounding without me being aware. So I'm just sort of, as a precaution, going to do these as float 64s, and nothing will really change by doing that, but it just ensures that everything's okay. It's good practice. And then what I want to make sure I do is I want to make sure that the input of the Find-maxima algorithm is scaled between 0 and 255. So it's just designed to work fast, and as part of that, the processing is done on an image between those limits. So I'm just going to take the distance array, make sure it's also in float, so, and then I'm just going to scale it between its max and min, like so. So that will now be between 0 and 1, and then I just times it by 255 to make sure that it's scaled now between 0 and 255. And then what we do is we calculate first the local max. So this is the function I had before at the top, and this is as an input for the Find-maxima algorithm, which basically prunes away the smaller peaks and joins them up. So the local max will find many peaks, and I'll show you that. And then the algorithm that I have will prune those and just bring out the more prominent ones based on the noise tolerance. So this is like a pre-processing, and then what we'll do is we'll just plot that data so you can see it. And I'll make it a bit bigger. So you can see there are many peaks here. Some of them represent proper maximums, and others are too similar to one another to really represent those maximas. So what we do now is we prune those away using the Find-maxima algorithm. And this has several outputs. It has the positions of the coordinates and also a representation of those spatially, which we won't actually use, but it's good to have. And then we're going to do Find-maxima, Find-maxima. We're going to put in our image, our distance transformation image. Just make sure it's in the right format with unsigned integer 8. It's between 0 and 255, so we won't lose any values doing it that way. And then great, so we've managed to run it. I might just get rid of that for now. So we've now got our coordinates of the maxima with y and x, and I can just actually plot those on the original just so you can see them. So we'll do plt plot, we'll do x and y, and do it as a red marker, and you can see that we've now nicely found the peaks of those. And then we want to use that for our watershed algorithm. Now, I've talked about how this works in another video, which I'll link to in the description. So you can see that below. And I talk about how it works, and I will link exactly to the seconds of the presentation where I did it, so you don't have to search for it. So what I want to do now is basically use this to mark the image. So I'm going to create an image called local maxi, which will be as an input for the watershed. And I want that to be the same size as our input. So do it, initialize it as a matrix or a 2D array of zeros. And then I'm just going to set where our coordinates are to 1. And then we're going to split our image like so. This will tell you how many there are and their location, which we can use later on. And now we're going to do the watershed based on this. So we do minus dist. I talk about that in the other video, why that's the case. We input our markers to show our seed points, and then we want it to explore our binary image and make that into a labeled image. So what I now will do is plot that. I'll try and do my subplots. Actually, I'll do it this way. I think that's the right way. Yeah. So we'll start with a distance one. And then second image. Can do the binary image. Also with the seed points on. And then we'll show our labelled ones, which is the most interesting, I think. And then we'll choose a lookup table, which will label each one a sort of alternate colour to make it easier to see where they change from one to the other. And figure is not, there we go, I spelt it wrong. Subplot. No, what am I doing? I just forgot the PLT. Still making mistakes. So actually we can comment this one out for now. We should have a nice row. Great. So you can see here on the right hand side, it hasn't done a perfect job of separating our cells, but it's done a much better job than it was before. So these cells, which were very close and sort of overlapping, are now separated. This is better than nothing, but it's not perfect. But generally this is good enough for getting a quantitative measure of what's happening. We could always exclude cells like this if we thought they were problematic, but as long as one condition isn't biased by the analysis compared to another, then it's all right to have some error, because all of imaging and analysis is an interpretation of what's really going on rather than the exact truth. So as long as your conditions are not being biased by your analysis in one way or the other, as long as it's kind of not perfect across the board and to the same extent, then it's still a good experiment. So we'll proceed with the analysis as it is. If you wanted to do this better, you could use machine learning to use something like cell dist or cell cognition, where they use machine learning to demarcate the cells, more powerful approaches. But I like to, for something like this, code it from scratch. But you can definitely use a more powerful approach. But this will be fine for now. So now we've been able to demarcate the cells, we want to actually count the foci in the other channels. And the way we're going to do that is, again, we're going to use this Find-maxima. So we're going to set the noise tolerance to be higher this time. And then we're going to set our input to be our channel 1 rather than the first channel, channel 0. And we're just going to... It's actually already in float, so I don't need to do that. And then we are gonna scale it between 255 and 0. So we just divide it by the maximum value and then times by 255. And then once again, we're going to find the local max. And then we're going to store the outputs that we want as something unique. See, I'm using the same variable names as before, which we've just overwritten, but that's fine because we're just using a lot of them temporarily, and I've given the outputs a unique value. Okay, that seems to have worked. So if I just plot quickly what that looks like. Just forgetting that PLT. So you can see this is where it's found the foci. It's perhaps not so easy to see, so what I'll do is I'll make it bigger and then I'll show you before and after. So let's make it red, try it again. So you can see that the brighter blobs have been now identified by the algorithm. You could make the value a bit lower here, noise tolerance, and you'll find more peaks. So if I were to do, say, 80, you can see it's found more, but it's also some of the more noisy ones. So there's a balance to be struck there. I'll go with 100 for this. So I think that's fair enough. And what I'll then do is I'll do it for the second image. So we can just copy this, or the last channel even, rather than the second image. So we're going to go with channel 2, and then just change these. These are the bits I need afterwards. And we can have a little look at this image as well. So This is a slightly less noisy image, and it's done a good job finding a lot of the foci. Again, the sensitivity can be changed with this noise tolerance, so you can make it lower. If you find that it's picking up a lot of noise, then you could filter the image with a Gaussian, but I'll keep it as it is for now. And then actually something I wanted to do, which I haven't done yet, is I wanted to populate a panda array so that we can start collating the data. So I'm actually going to go back up above, and I'm going to say, before the plotting, I want to create a data frame in pandas, and from our data, and the data will look something like this. So to do this, I'm basically defining the columns. So we're going to have one column called image, and this is the image name here really. So what I'll do is I'll say image name is this. And the reason I'm taking this, splitting this out now, is so that you could write a script which cycles through the image names, and everything else is set. So the channels are discrete from the bulk of the image name, and this is sort of the root which is consistent. So we'll take that, we'll run that, sorry. So I forgot to add in the directory when I took that out. Like so. And then in our data frame we'll have our image name to start with, and then I want to have our cell number, because each image has a number of cells, and this was extracted when I created my Find-maxima label here, num, so we'll do 0 between num. And then I want to record the location of the foci in each channel. So I haven't got the values for that yet. I'll do that below. But I'll just make the columns now and populate them with none. And then we'll run that, see if it works. Pandas has no attribute called data frame. That doesn't sound right, does it? Set a capital F maybe. Yep, so that looks like it's worked. We can actually have a sneaky peek at our data frame here, and yeah, you can see that now we've got a row for every cell that's in there, which is prefixed with the image name, and then we've got space for all the points. So now we've identified the points, we'll put them into our data frame. So we'll do that as an array. We'll add two arrays basically, which we'll make first of all. So create two empty arrays, and then we want to go from each cell and create an array within an array, so that we can keep them separate. So we don't want just one long array for each one, we want a separate array for each cell, and then we will go through the points in our overall image foci detection and see if they fit in the cell. So the way that we can do that is to create a new image which equals wherever our label equals a certain value. So our labeled image has each cell, each spatial location of each cell represented by a number. So one cell will be all ones as a mask for that cell. The next cell will be all twos and all threes. And so we say where labels equals 1, 2, 3, then that will be where the cell is, which is represented as this bint here. And then what we say is if for a certain foci, and we run through the list of the foci that we've detected multiple times, if it equals 1, then we can say that that foci belongs to this particular cell. Like so. So yeah. So if it equals, if it's in that cell, then we want to actually add it to the list. So that will go through in the first channel. It will find if a particular foci overlaps with a cell, and then it will add it to the sublist of the main list. And then we want to repeat that for the foci from the second. Sorry, that's very confusing. So this zip just means I can unravel these two lists at the same time. So it will take a value from each list one at a time. So you'll get a Y and an X in one round, and then the next Y and X in the next round. So it kind of unravels them simultaneously, one by one, which is very convenient. And then we'll check if these exist as well. Within a cell. And then if they do, then we'll add it to our second list. Do I actually, yeah, that's it. So I'll show you what these lists look like. It should be more convincing. Okay, so I've just mistakenly done the wrong type of brackets there. Should be square brackets. And then I'll show you what that looks like. Yeah, so this will be the first cell. So we've got two foci. This is the second cell. We've got two foci with x, y coordinates. And then some cells have more than others. And then if we compare this to the second channel, we can see that there's different coordinates, some with different numbers of foci there. Okay, so that should be fine. And then what we want to do is make sure we add this to our data frame. But actually, I think I just want to check what labels equals 0 looks like. That might be the background. Yeah, that's the background. So actually, we want to start with a +1 like that. So the cells actually in the image are labelled from 1 and up, like that. But the number of images, the number of cells starts as though it's from 0. So that will work. So now we want to populate our data frame with this information. And so we just set it like this. Like this. And we can have a little look at our data frame, and you can see now we have indeed populated each one of our cells, like so. So yeah, you see here we've got cell number starting from 0. It doesn't actually correspond to the actual value in the image, so I think what I will do, just to make it more kind of simple, is to actually do that so it does. So here we'll have between 1 and +1. So now when I look at the data frame, this goes between 1 and 27, which is just the values that we actually have in the label image, so everything corresponds. But I can see I've actually made a mistake here. It's not a big deal, but that should be PTS. So I populated it and then added 2 more columns, which was a mistake. So yeah, now we've got a nice compact data frame. And this just makes sure that our data is nice and tidy so that we could export it to Excel or something like that. You don't need to use data frames for this kind of processing, but it can help just keep things tidy, especially when you're analysing lots of cells. But the final bit of the analysis is to establish whether our cells are actually co-localized. So the foci are actually co-localized within the cells. So what we'll do is we'll create another empty array which we're going to populate, co-loc number, so this is the number which are co-localizing in each case, and then we're going to work for each of our cells in range 1 num plus 1. So we're actually going to index specifically, and then we want to go through our data frame, and we want our data frame where the cell number equals a certain value. So this just makes sure we're really getting the right cell number, and we're going to say equals I, and then we want the values not from cell num, that would just be the number that we've searched for. We want to actually find the value of the corresponding foci locations, and I'm pretty sure we will have to index it like this after making it a list like this. So if we don't do that, if I just try that once, so yeah, that's what we want. If I did that, you could see that it's actually still in brackets, so the 0 there just makes sure that we've got an array without too many brackets on the edge, parentheses, and then we want to do the same for our second list for the same corresponding foci for the same corresponding cell. And then we want to do a very simple co-localization by measuring the distance. So we want to know how many are co-localizing and then add to that if we find any that are. So go through each list and we'll compare the coordinates of the foci. So you could do this with a numpy array, compare the dists, but there's such a small number really, we can do it this way, which is a bit more explanatory. So we're going to basically measure the Euclidean distance, or it's like the hypotenuse on a triangle. So you want to compare the difference between the x's and square it, and then compare the distances in the y's. And square it so it's positive distance, and then the square root basically puts it back into the space of the original measurements. So we subtract the x's, square them, plus the y's, square them, and then calculate the square root, and that's our dist. And then we basically want to come up with an arbitrary value for what represents the co-localization. So if they're within 10 pixels of each other, then we say that they, in this case, are co-localized. But this value can be changed if we find that it's too strict, and there might be some chromatic aberration or something which means that they're a bit further apart. You could make that higher, or you could even make it smaller if you feel like you're being too relaxed with your definition for the colocalization. And then whatever our count is for a particular cell and how many that's co-localized, we add that to our overall array. It might be 0, but probably be more if they're closer, and then we can add that to our data frame like so that we've got all of our data nicely arranged. And that looks like it's worked first time. So if we do our data frame, we can see now that we've got some counts, but it would be nice just to also visualize what's happening a bit. So if we let's once again visualize our first channel, and then on top of that we can actually plot our detected points. So I think that's right. CH1, CH1. We'll make these ones red, and we'll set the alpha so if they're overlapping we can actually see that, and we'll make them quite big so that we can see them on the image. So marker size equals, say, 25. It's just for visualization. And then we'll set this to, say, cyan for anyone that's colorblind. Oh wow, what did I do wrong? Oh yeah, okay, I've just forgotten. That's very large, isn't it? But it's because I haven't scaled the figure as well. I'm sure I could set this globally, but it's good practice to type it in. Yeah, so now you can see where the foci are overlapping. Cyan really wasn't a good choice, but maybe we could make that black, if that can be better. Yeah, well, you can kind of see where they're overlapping and where they're not. So that's it basically. And yeah, your data frame has all of your values in, so you could then repeat this for other cells and collate them to the same data frame, working through each one and creating functions out of these so that they can just be called. And that's a kind of very quick way to analyze foci in individual cells and compare the distance, and then come up with an arbitrary check for whether they are co-localized or not. So there are many different ways of appreciating co-localization. This is sort of the simplest form, and it doesn't really take into account if you've got multiple foci within range of an individual foci. For example, they'll be counted more than once potentially, but this is sort of a good starting point. You can now go off and refine the algorithm and see if there's, a way of doing it which is perhaps more true to the biological question. but there are many ways of interpreting what's truer. So this is sort of the most basic level, and you can take it from here for a more refined analysis. Okay, perfect. So again, this will be linked to in the description for the video, the GitHub with this page and all of the requirements, and I will also link to descriptions of the segmentation and also the Find-maxima algorithm in the description for the video. Thanks very much.